So next in our macromolecule list are proteins, or these can also sometimes be called polypeptides. So these two terms are interchangeable. So the monomer of this group is called an amino acid. And an amino acid is going to start off with 4 different parts. The first part is going to be our central hydrocarbon. Here's our carbon molecule. Here is our hydrogen. And so you can see we can still make 1, 2, 3 connections with our carbon molecule. One group that we'll put into one of those is that carboxyl group. So we talked about the carboxyl group when we talked about functional groups. This is one carbon with 2 oxygens and a hydrogen, COOH. So you can see it has one connection it's going to make, and so it will make one connection with that central hydrocarbon. We still have 2 more connections we can make. Another functional group that we talked about was the amino group. A nitrogen with 2 hydrogens. So the nitrogen is the blue, the hydrogens are the white marbles. They will make one connection, so we can see we have one connection we can make with that, and so it will go into the other side. Now we can make one more connection. This is the unique part of every amino acid. Every amino acid will have these 3 same parts: an amino, a central hydrocarbon, and a carboxyl group. The part that will change, and there are 20 different types of amino acids, so since it's going to change, we're just going to represent it with this flipper. So in the 4th hole for our carbon, we will put the R group. And you can look in figure 317, and you can see that they have drawn out 4 different amino acids. And in those, you can see that the amino group always stays the same, the NH2. The carboxyl group always stays the same in all 4, a hydrogen coming off of the carbon. But then in the purple box, you can see that the R group, that is what is changed for each amino acid. So that is our basic building block. That is our monomer for proteins. So I need a little bit of room, so I'm going to erase those 4 things. So if I put together a string of amino acids, I will create a protein or a polypeptide. So the name of the polymer given to proteins is also the name of the group. So proteins or polypeptides are the names of the polymers. So remember, if we're going to build, we're going to do a dehydration synthesis. So here I have one amino acid. In dehydration synthesis, we take the hydroxyl group, a hydrogen and an oxygen, off of one amino acid, and we take a hydrogen off of the amino group, and now they can form a bond. So the nitrogen is looking for 1, it will form a bond, and it will form a bond with that carbon where we took that hydroxyl off. Now we have built ourselves a protein. This would be a polypeptide because we have more than one amino acid, poly, and peptide is because the bond that we just created between them is called a peptide bond. So the molecule is called a polypeptide because it has many peptide bonds. We could keep doing that. We keep doing dehydration synthesis, and we could keep building them. Or we could do hydrolysis, in which case we would break our peptide bond, and we would put our hydrogen back on our amino group, and we would put our hydroxide group back on here, and we would be back to our original monomers, the amino acids. You can see that example also in figure 318. So remember, if you go one direction, if you have an amino acid plus an amino acid, this would become a protein plus that molecule of water. But backwards, so this is synthesis, you go the other way, protein plus that water would become an amino acid and another amino acid. So when you build and break these molecules through synthesis and hydrolysis, they just go back and forth. Building, synthesis, if you synthesize, hydrolysis, breaking. So we break that big polymer into its component amino acids. All right, so those are the basics of our monomers and polymers. For proteins, we also talk about structure. A protein or a polypeptide has many different forms before it becomes functional. So, as we begin to do synthesis, we begin to build a chain of amino acids. We keep doing dehydration synthesis, and we keep bonding our molecules. All those dashes are representing the peptide bonds. If we were just to list all of the amino acids in a row, that would be its primary structure. Or you could use the symbol 1 degrees. So this is also in figure 319. All right, so that's the primary structure. This is not a functional protein though. The different functional groups of those R groups, however, are going to start interacting with each other, and we'll begin to build the secondary structure of a protein, or 2 degrees. So some of those amino acids are going to come together, and they're going to start making these what are called beta pleated sheets. It looks like a skirt, like a pleat. And that's going to depend on what kind of functional groups are there. Other parts of this chain are going to begin to make curlicues. These are called alpha helices. A helix is a spiral, like a pigtail. And so different parts will do pleated sheets, different parts will do squigglies, alpha helices. So these two things are what we call the secondary structure. This is still not a functional protein. It's not until it folds up into a 3D or tertiary structure. Or another word for tertiary, this is called its conformation. This is the functional protein. It's not until it gets into this shape that it can do its job. And so you'll often see these, and you see this in the picture where these curlicues and these pleated sheets are all folding over onto each other, and it makes it a bit of a mess. But we went from a linear structure to a 3-dimensional structure, or I should actually say a linear to a 2-dimensional to a 3-dimensional. We now have our functional protein. Now there's one more type of structure. It's called quaternary. And quaternary just means you have more than one protein in its tertiary structure. So if we were to make one protein, as we did, so this is the tertiary structure of one protein. So I'll just make a little bit of a mess. So that's one protein in its tertiary structure. We could also have another protein in its tertiary structure. So we'll put some pleated sheets in, we'll put some alpha helices. So this is, the black is one protein in its tertiary structure. The red is one protein in its tertiary structure. Combined, now that they're working together, that is the quaternary structure, or 4 degrees. This is 2 or more proteins in their tertiary structure working working together. In the figure on 319, they actually show 3 proteins working together. But they are 3 independent proteins. Their combination is what makes the quaternary structure. All right, so that gets us proteins. The other thing I've been wanting you to do is to know their functions. So if you look at the beginning of this section, they talk about all the different functions. And we can break up functions into 2 groups. Metabolic, remember we have that term metabolism. These are the different chemical equations that are happening. So your proteins are involved in making sure all those chemical reactions work, as well as being structural. So the biggest one for metabolism is enzymes. Sometimes in those reactions, you have your reactants, and you still have your products, but sometimes an enzyme is going to help. Actually, most of the time, an enzyme is going to help. And we'll talk more about enzymes in chapter 6. But right now, just know that an enzyme is another molecule, a protein molecule, that will facilitate our chemical reactions. They can also be used for transport. Remember, in the plasma membrane, the membrane was mostly composed of phospholipids, but there are proteins in that plasma membrane that also allow things to come into and out of the cell. Another one, antibodies. You've been hearing a lot about antibodies in the news. Antibodies are proteins that are created by your immune system that can help to fight different diseases. This is what we look for to see if someone has had COVID-13, because if you had it, your body would have created an antibody in order to fight it. And then they can also be for regulation. We talked about how steroids are hormones, these chemical communicators. Some of your hormones are lipids, steroids. Some of them are protein-based. As far as structure is concerned, a good portion of your body is muscle, and muscle is protein. So muscle, hair, your fingernails, these are all composed of protein. Or another one that's very important, blood. Blood is a protein. Or, I should say this is number 1 under structure, or 2, it can be used for motion. Your muscles, remember, are what move you. So your muscles are attached to your bones, and as they contract and relax, they allow the movement of bones, which allows you to have your motion. So these are the different functions of our proteins.